उम्मीद है जी आप सब खैर आफियत से होंगे. So now let's start the discussion in detail that how a normal cell converts into a cancer cell. So in a recent lecture we have discussed about the mutations, the gain of and loss of mutation, the concept of mutation, and we have stopped our discussion up to this point, that we first understand the normal cell cycle pattern. And very briefly, I am not going into a detail of the every gene, but just suppose if this is a cell, and of course it is its nucleus, and in the nucleus there is DNA. Okay, let me change the color. This is the DNA, you can say. Fine. Okay, so there are whole group of genes. Just suppose these are the one group of genes, these are the another group of genes, and these are another group of genes. Fine. This gene is responsible for, whenever this gene will be stimulated, the cell undergoes the proliferation. So this is responsible for synthesis. For the cell proliferation or the cell cycle, the crucial step is the synthesis. What synthesis? The DNA synthesis, the replication in the S phase. Understand? Or whenever, if this is the stimulated, and whenever this gene would be stopped, the cell undergoes, or you can say the stop proliferation or stop synthesis, isn't it? So this gene is under the influence of what? These are the two other group of genes. This gene is making a product, and this gene stimulate this one. If this one stimulated by this group of genes, cell undergo cell cycle. So it's mean these are the accelerators, and whenever this gene will be activated, it will stop this gene. So this act as the brakes, you can say, and this act as accelerators, isn't it? So these are the actually proto-oncogenes, and these are the tumor suppressor genes. This is tumor suppressor gene, and this is proto-oncogene. So proto-oncogenes are the cell cycle accelerators, and tumor suppressor genes are the cell cycle brakes. Whenever the POG genes, you can say proto-oncogenes, are stimulated, they will stimulate the group of genes, these group of genes, and the cell undergo proliferation. And whenever the tumor suppressor genes will be activated, they start to resist the cell cycle, or they start to resist the proliferation, and they will inhibit this group of genes, and the cell cycle stops. This is what normal cell cycles going on. Fine. So now, to verily become a cancer cell, or when a normal cell convert into a cancer cell, so what kind of mutations over there? Either the gain of function or loss of function. Like first we talk about the proto-oncogenes. Proto-oncogenes getting what kind of mutations? If you say the proto-oncogenes getting loss of function, so there is no cancer, because we know for the cancer the cell must be over proliferate. There could be the abnormal cell cycle, or you can say the uncontrolled cell division. So proto-oncogenes must be getting gain of function, and tumor suppressor gene must be getting loss of function. This is important, and I hope you understand. If tumor suppressor genes getting the gain of functions, so of course there could be no any cancer, because it is over proliferating. It is something like a you can say the rock, and it will plays its role more appropriately in an enhanced manner, or you can say the very powerfully stimulant, as a stimulant to stop the cell cycle. But to verily, to you can say the to become a car, really an abnormal car, or you can say the out of control car, we must enhance, or we must accelerate over, and we must fail the brakes. So At that time and in that situation, the cell or a car, if analogy you take as a car, so and car become really a, you can say, a out of control car or abnormal car. The same true is about the cell, that you have to over accelerate it and under brake it, isn't it? So this is one thing. So abnormal cell, you can say the normal cell, after getting the mutations in such a way as I have mentioned, so cell become over proliferate and ultimately it will lead to the malignancy. But you can say how over proliferations over there, like how POG genes getting the okay, so in a normal cell cycle all these genes are under the particular cues, particular signals. But whenever there is a mutation, so might be possible, if there is no any signal from POG, if there is no any signal in the POG, might be possible this gene is overworking automatically, and this cell now become deaf to the tumor suppressor genes. There is no any obeying to the inhibitory signals. So we can say now the cell is becoming self-sufficient, self-sufficient in its growth, isn't it? Or you can say the cell is becoming autonomous. So this is the first condition in the malignancy, or one you can say the difference, that a normal cell cycle is under the strict control, but a cancer cell is self-sufficient in their growth mechanism, or you can say it is not obeying any particular signals, or you can say the any growth signals or the stimulatory signals. It divides or it proliferates on its own automatically. So this is the first thing. Now continue our discussion ahead. This is not the easy case that you just say that okay fine, to damage the genes related to the cell cycle is enough to blow a normal cell into a malignant cell. Okay, cell cycle, fine. Now another principle, what normal behaviors, what is the normal behavior? First we have understand the normal, then we can understand the malignant automatically. The normal behavior is this, that whenever a DNA is damages, whenever a cell DNA is damages, just suppose, again taking our original diagram, these are the DNA. So these are the group of genes, that particular proton genes and growth genes and the tumor suppressor genes. Whenever the damage to the DNA, not in the normal cell, another group of genes are there and they will activate. They will, you can say, the activate. And what they will do? They will open a toolbox and just do repair these genes. What does it mean? That in a normal cell, whenever a cell undergoes a damage, whenever a cell's DNA undergoes the damage, so there are the repair genes are there, so it will try their best to repair the DNA. It is the normal cell mechanism. Now just suppose another mutation is over there in this group of genes, the cell repairing genes, or you can say the cell beneficiary genes. So in this gene group of genes, what kind of mutation over there? Either the gain of function or the loss of function. So of course there could be the loss of function. Then it's mean there is another mutation plus repair genes. Repair genes are also undergoes the mutations. You will see that how much mutations over there in a particular malignant cell. There is not a normal damage or normal 2 to 3 mutations, so then cell become a malignant. No, not in this case. But to become really a normal into a malignant, we must have to become a damage, to done a lot big damage to the DNA. So first of all, there is a cell cycle genes damage, and then top of it, the repair genes are also mutated, so that they cannot function well. Okay, now another thing, that whenever, now we will see the normal function. When a cell realizes that okay, my dead genes are also damaged, fine. Now it will realizes that there could be the repair genes that can also not function there well, so these are also damaged. Then another group of genes, they are related to the apoptosis gene. They are related to the apoptosis. Actually, there are the two genes. Just pause. If this gene this gene is for, you can say, the anti-apoptosis and pro-apoptosis. Actually, these are the two genes, anti-apoptotic genes and the pro-apoptotic genes. Anti-apoptotic genes are resisting the apoptosis, or it means they are cell survival genes. Whenever they are activated, cell become a long age, or you can say the long live. And when the pro-apoptosis gene will be activated, cell undergoes the apoptosis. So these are the two kind of genes, anti-apoptotic gene and pro-apoptotic gene. Now you have to tell me, okay, first one I will tell you that in a normal cell, when cell realizes that okay, I have a lot of damage to my DNA, so cell realizes that this particular damaged DNA should not pass to the next progenitor cell, or sorry, next daughter cell. Cell undergo apoptosis. So this gene will be stopped and this gene will be on. It is normal. But in cancer cell, you will think that what could be the scenario, that anti-apoptotic genes will get what kind of mutation? Anti-apoptotic gene will get the gain of function, and pro-apoptotic genes will gain the loss of function. So cell does not undergoes the apoptosis. So cell become overlived, over survived. Understand? This is the another implication of the cancer cell, or you can say the mutation. So another mutation now you have in your record, that the apoptotic genes are also getting the mutations. The anti-apoptotic genes are getting the gain of function mutation, and pro-apoptotic genes getting the loss of function. This is a third difference. Now, okay, now another thing, we know that whenever cell replicates, there are something like chromosomes, and these are the telomeric ends, isn't it? Whenever cell replicates, the chromosomes are shortening. This is the basic principle. I am not getting in detail about all these things, but I'm just this lecture is just for the difference between the normal and abnormal. So in every replicative cycle, the chromosomes become shortened. So according to this principle, no any cell will become a cancer cell, because we know the normal cell become a cancer when it is over proliferate, and when it is over proliferate, of course the chromosome must be shortened, and at the time is reaches that the chromosome essential DNA genes would be damaged and the cell will die. But this is not happening. Actually, what is there? Another gene is there. Another group of gene is there. It will synthesize an enzyme, and that enzyme is telomerase. There are many normally, actually, in a normal cells in our body, there are many cells that are long lived. So how they are long lived? Actually, in that cells there is an enzyme that is telomerase. It will adding the telomeres on chromosomes after every replicative cycle, so cell become more long lived, you can say. And some cells have the short lived, so telomeric activity is short, you can say. So in a cancer cell, when cell is get mutations, cell also getting the mutation of the dead gene that is responsible to form a telomerase enzyme. So what kind of active and mutations over there? The telomerase related enzymes are getting the gain of function, so the telomerase activity is enhanced. So there is a no more shortening of the chromosome, rather chromosomes are maintained at their level, so there is no any damage to the DNA. Rather, a cell is becoming more and more and more abnormal DNA and converting into a cancer cell. This is another, you can say, another mutations. The telomerase are also mutated. Now you can see we have damage to the proto-oncogenes, we have damage to the tumor suppressor genes, we have damage to the repair genes, we have damage to the apoptotic genes, and apoptotic, they are the two DNA groups, the anti-apoptotic and pro-apoptotic. We have also damaged the telomerase related genes. These are the almost 5 to 7 mutations we have done at that time. Now sometimes normally cell physiologically live long actually, but there is an enzyme that is telomerase. So if this is mutated abnormally and function override, you can say, so the cell will survive along. On the top of it, there is a anti-apoptotic that are mutated gain of functions, and more of it that the telomerase are not shortening well. So you can say this thing, the telomerase activity, what is this? This is the limitless, you can say, the limitless replicative capacity. This is another feature of the cancer cell. Now some of them are also over there. You know, the every tissue in our cell or in our body, sorry, not cell, in our body, they need nutrients. They need nutrients because they have to survive. So for this there must be a, what is this, channel? No, this is a vessel. So there must be a vessels over there, so they can, so blood can reach over there and they will transport the nutrients and removes the waste as well. So a malignant cell has such a you can say the mechanism that it will generate some chemicals, or you can say signals, that is promoting the angiogenesis. So the more vessels are formed around the neoplastic, you can say, growth. Like just suppose if this is a neoplastic means new growth, or you can say abnormal growth. If there is a tumor is forming over there, so it will release such kind of substances that the blood vessels, if blood vessels are going over there, so more blood vessels are coming out of it, and the more blood vessels round over over this structure, so they will sustain and they will maintain its, you can say, the nutritive and the excretory activities as well. So it is the angiogenesis, another feature of the malignant cell, that it is promoting the angiogenesis. Up to now we have damaged the DNA, a lot of the damage. We have damaged the cell cycle genes, repair genes, apoptotic genes, telomeric genes, or you can say the, we have also sustained the angiogenesis. But up to now a normal cell is not becoming a cancer cell. Might be possible it is becoming a benign tumor, but not a cancer cell. To be really a cancer cell, there must be a one another properties over there, and that is the metastasis, to invade a tissue. This is important. This, I think, it would be our last thing to discuss. And please concentrate with your both ears. Everyone has both ears. First I will see the, I will tell you the normal cell behavior. Again, take a skin piece. If this is a skin cell, of course there is a, you can say, what is this, dermis. If this is epidermis, this is dermis. Do you think this epidermal cell can go towards the dermis? No. Actually, what happens, our normal cells are not dislocate or leave their anatomical confines. They live happily where they are. This is important thing. This is you what have to understand. Actually, every normal cell function its well in its own area. Like if I put my one liver cell into my brain, so there will be a mini liver. No, not of course. Actually, it will die. The liver cell will die. So, what kind of the behavior is this? Because our every cell knows its own parenchyma, its own surroundings. They talk to each other. Okay, I am in a appropriate environment. Yes, I am in a you can say the environment where I have to be or I supposed to be. So our cells have a recognition patterns. Our cells knows where we have to be placed, or they cannot cross this boundary wall. So they are the two things that normally if this cell by any mean passes over there, it will die over there. But how when a cancer cell is becoming and will travel here and there, so how it will survive? This is one thing. Second thing, normally a normal cell can never leave its boundary due to some anatomical confinements, or it has not such kind of enzymes that will break down the proteins and basement membranes and then invade or pin their nose to another areas. This is not such a case in a normal cell. So there are the two more things that in a cancer cell that is called invasion to the other tissues, that is particularly called metastasis. So cancer cell another property is metastasize. And how it is metastasize? Actually it will open up those genes that are embryonically actively genes. Embryonically active genes mean, you know, when the embryo is formed, initially it is a zygote, and when it becomes the two cell stage and four and so forth, and becoming a whole organism. So what is this? As the time passes, whenever the cell are increasing in their number, they become more specialized, or we can say they become differentiated. So whenever the cell become differentiated, although just suppose if this is a differentiated cell, it has also 46 chromosome, it has also 46 chromosomes, and if you take the chromosome number 1 and if we take its chromosome number 1, here all the genes are active, but here not all the genes are active, because whenever cell become differentiated, some of the genes become switch off and some of the genes become on. Because if the genes are no more any, you can say the in need, so it is off. But in a zygotic life or early embryo life, almost all the genes are activated. So cell is undifferentiated. So undifferentiated cell can be go here and there and it can survive. So abnormal, so cancer cell has a more feature that it will open up that genes that are embryonically active genes, or you can say it will de-differentiate itself. It is another property of the cancer cell that it will de-differentiate. This is important thing. And moreover, cancer cell has such kind of enzymes, it will produce such kind of enzymes that will cut these boundaries and they will metastasize. So this is the, you can say, how a normal cell has the a long journey to become a cancer cell. Now the last thing, our last punch, you know, we have succeeded to become a normal into a abnormal, isn't it? So do you think, we know that a normal cell produces normal messenger RNA, normal messenger RNA produces normal proteins, and normal proteins, some of the proteins are excreted proteins and some of the proteins are the, you can say, the embedded in the cell membrane, and these normal proteins are recognized by my immune system. So whenever our cell passes, immune cell our passes by my all the tissues, they will all the time keep an eye on every cell that either they are normal, either they are my cells or self, or either is there any foreign substance. So what do you think that if a cell become abnormal, its genome become abnormal, so of course it will producing the abnormal mRNA, and abnormal mRNA will translate into an abnormal protein. So some of the abnormal protein will be displayed or presented on the cell membrane. So whenever my immune cell passes over there, it will recognize it, isn't it, and damage it. But again, it is not the case, because cancer cell has also the ability to escape the immunity. It is, you can say, the makeover, make it up in such a way that it masks the effect of the immunity. So immunity cannot, you can say, the overcome this situation. So this is the whole in a nutshell, that what we have done. We have damaged cell cycle. Cell becomes self sufficient. Top of it, cell cannot repair itself. More of it, cell has the increase the replicative capacity. Top of it, cell, you can say, um, the you can say the sustained angiogenesis. And then cell has the ability to de-differentiate. Then cell has the ability to break the boundaries and escape immunity. So this is the whole, a lot of damage to the DNA, and then a normal, innocent cell is becoming a abnormal or cancer or malignant cell. So this is the basic difference, that how a normal convert into the malignant. But there is a lot of discussions, a long lecture over there, that how, even if we talk on the cell cycle related genes, like you can say the proton cogenes and tumor suppressor genes, I think that could be a lecture about 1 to 2 hours only on this situation. So I hope it is clear to you. If you have any questions, just do let me know on comment section. Take care of yourself. Allah Hafiz.